I'm going to solve the May 2017 EDEXCEL question paper 1b. So I had solved the question paper and I had solved it with answers. If you check the answer, and the answers had been given elaborately, all the answers have been given. So if you need the answers, you can check the answers whenever you at the time of solving question paper. Thank you. Here I am going to start today biology EDEXCEL paper 1b May 2017. So let's start. First, question number 1. The diagram shows part of the human breathing system. So there had been shown one breathing system. Name the structures labeled A, B and C. So we know A is trachea, or we can say windpipe, B bronchus or bronchi, and C it is bronchioles. Number 2. Suggest why the lung structure is sometimes referred to as a tree. It is for the branches. It is of the branches which seen in the diagram. We can, for this reason, these branches referred to as a tree. So you just, if you say that for the branches, and you will get the full marks. Next question, number B. Describe how ventilation of the lungs occurs when a person breathes in itself. That means they wanted to know the inhalation. So what happens when inhalation or a person breathes in? When the person breathes in, the diaphragm contracts, pulling downwards, and the intercostal muscle contract, expanding the rib cage. So the volume inside the thorax increases, which reduces the pressure in the chest, and as a result air moves into the lungs. These are the process. So there are some processes. First you have to indicate about the diaphragm, then you have to indicate about the intercostal muscle, and we know that there are two kinds of intercostal muscle. One is internal intercostal muscle, other one is external intercostal muscle. If you just say about the intercostal, you don't have to indicate about internal and external intercostal muscle. You just say only intercostal muscle. Diaphragm, intercostal muscle, thorax and pressure. These 4 points are very important, and for this reason they had given 4. Now coming to the next question. Smoking cigarettes inside public buildings has been banned in many countries. Suggest why governments have banned smoking cigarettes inside public buildings. Because it affects non-smokers as well. This damages their health against their will and could cause them cancer. Tobacco smoke contains many harmful substances like tar, nicotine, etc. Suggest why children are particularly at risk from breathing in smoke from other people's cigarette. It is for the passive smoking. and as they are children, they can easily catch the smoke, which may develop cancer cell, and as a result they may have a lung cancer. Different kinds of problems may occur like asthma, bronchitis, and there is all children are smaller, lungs are smaller. Children and teens who smoke are affected by the same health problems that affect adults. Secondhand smoke may cause problems for children later in life, including poor lung development, meaning that their lungs never grow to their full potential, and later it may cause also lung cancer. Number 2, a student investigates the movement of substances from the gut into the blood using a model. The whisking tubing bag represents the gut and water represents the blood. Whisking tubing is permeable to small molecules. So here they are telling whisking tubing is a bag, and in the bag there are some substances, and surrounding the whisking tubing is a water. Now the concentration of water is more inside the bag, definitely, and it is covered with plastic cover. The student puts different solutions of various substances into the bag. That means into the bag the student gave different kinds of solutions. After 20 minutes he carried out food test. Complete the table to show the results obtained by the student. One has been done for you. So here you see, when we see the starch, only starch in the bag. In the water there is no starch, in the glucose also, in glucose column also there is no, because starch is a large molecules. It cannot pass through the whisking tubing. So it will remain in the bag, and the water, and in the water there will after starch breaks down it convert to glucose, but starch is not able to break down as there is no enzyme, nothing. Now second column, there is starch and glucose. So here after the substances in the bag, so solutions in bag, we are seeing that then it is present starch and glucose. In the glucose column it will be all number, in glucose column it will be yes, in glucose column it will be yes for starch and glucose, and starch present in water, starch and glucose, it will be no, as starch is a big molecule, it cannot pass through the tubing to the water. In case of starch and maltase, in glucose column it will be no. So in case of starch and maltase, in glucose column, if glucose present in water, if there is no, automatically it will be no in starch present in water in case of starch and maltase, because if starch breaks down then it will convert to maltase or glucose. Here as it is saying it is maltase, but starch didn't break down, that means it cannot go into the water. So in starch present in water it will be no in case of starch and maltase, and glucose present in water it will also be no. The next, starch and boiled and amylase. So boiled amylase have no function as it is boiled, the enzyme is already denatured. It cannot break down starch. So in both the columns, in starch present in water it will be no, and in glucose in water it will also be no. So I think the question is clear to you all. So let's go to the next question. Explain how the volume of the bag would change when it only contains starch solution. What we are seeing over here is that they told explain how the volume of the bag would change when it only contains starch solution. So the answer is there's a high concentration of water in the surrounding solution than inside the bag. The concentration of water is more which are present inside the bag. So water will move to the bag by osmosis, by a process called osmosis, and therefore the volume of bag would increase. So we know what is osmosis. Osmosis is a process where the water will move from high concentration to the low concentration. Here water which surrounds the bag is in high concentration, and inside the bag the water have low concentration. So there a osmotic gradient is produced. So water will move from the surrounding to the bag, which will increase the volume of the bag. Now next question, number 3. Describe the test the student should use to find out if glucose is present in the water. Number 3. Describe the test the student should use to find out if glucose is present in the water. So the answer for this is, in hot water, bendic solution gradually turns from blue to cloudy or orange or brick red when heated with a reducing sugar such as glucose. So we have to give the heat in hot water. The experiment should be done in the hot water. Now go for the next question. Explain how you could use this apparatus to investigate the effect of bile on the digestion of lipid. Here bile, lipid and lipase could be set up in one bag, and in another bag it will be only lipid and lipase. Digestion could be judged by testing the water for fatty acids and glycerol. So in one bag, whenever we are giving bile, lipid and lipase, there will be a reaction and the lipid will be broken down and it will form fatty acids and glycerol. It will then transfer from bag to the water. And in another bag, it will be lipid and lipase. There will be no bile. And over there also, as lipase is an enzyme, it will break down lipid into fatty acids and glycerol. Now in the water we will be found fatty acids and glycerol. So digestion could be judged by testing the water for fatty acids and glycerol using a pH meter. So we can measure the pH meter or suitable indicator in the water. In water, how much fatty acids and glycerol found, we can measure it by the suitable indicator or pH meter. And also how quickly fatty acids are being produced at a particular pH. For these experiments, for these different experiments at a particular pH, how much fatty acids, how much lipids are broken down and converted to fatty acids and glycerol, we can find out by this experiment. Next question, number 3. Galactosemia is an inherited condition. It is caused by a mutation to a gene on chromosome 9. The gene codes for an enzyme that removes the sugar galactose from the body. That means this gene codes for an enzyme. In chromosome 9 there is a mutation of gene. This gene codes for an enzyme that removes the sugar galactose. Normal allele capital G can produce the enzyme and is dominant. Recessive allele small letter g. The pedigree shows the inheritance of a galactosemia in a family. Now here we are seeing the unaffected male, unaffected female, male with galactosemia, female with galactosemia. Now number question A1, how many individuals in the family pedigree have an X and Y chromosome in each of their body cells? Now they say how many individuals have an X and Y chromosome. So we know that male contains X and Y chromosome, female contains X and X chromosome. So let's count all the boxes, square boxes over here. How many boxes? 1, 2, 3, 4, 5, 6, 7, 8. 8 square boxes. So the answer will be 8. And all of these are male because male contains XY chromosome. Now number 2, how many individuals in the family pedigree are homozygous recessive? Now here in this sentence already told, if you look at the sentence, the recessive allele is small letter g. Now by seeing the diagram we can easily understand those who are affected, they all will contain the homozygous recessive allele. Only galactosemia will happen to those who have recessive homozygous recessive allele small letter g. So in this diagram how many we are finding? 1, 2, 3, 4. So the answer will be 4, homozygous recessive. Number 3, give the genotype of individual 1. So here they say the genotype of individual 1. Now it will be capital G and small letter g. Here in the diagram we see that there the white boxes are unaffected. If unaffected when? If there is a capital letter G. So in the box it is unaffected male. In order to have this disease we need 2 recessive allele small letter g. So it is unaffected, that means there will be 1 capital letter G. Now here if you see the diagram properly, I am showing you genotype individual 1. You see that 1 and 2, but their children is affected. Here their children is affected, that means they have a recessive allele G, and also that means number 1 individual, it contains heterozygous. It is heterozygous and it will be capital letter G and small letter g. And if you also see in the next diagram of 8 and 9, you see the children are not affected. Okay, that means 8, it is also heterozygous, 9, it is also heterozygous, and for this reason the children are not affected. That means there is dominant capital letter G in both 8 and 9. Now if you see the next question, individuals 8 and 9 are going to have another child. Give the probability of this child having galactosemia. 3a, number 3. Here it is written, give the genotype of individual 1. So the genotype of individual 1, it will be capital letter G and small letter g. Why? Because we are seeing that here their children are affected. So if the genotype in case that if it is capital letter G and capital letter G, then the children won't be affected if both of them are dominant allele. But here I am seeing that the children of 1 and 2 are affected in number 5. That means the genotype of 1 it will be 1 is dominant allele and the other one is recessive allele. 1 it will be capital letter G and the other one is small letter g. Number 4, individuals 8 and 9 are going to have another child. Give the probability of this child having galactosemia. So in 1 we saw that it is hetero. Now in the next question they already asked the probability of this child having galactosemia. That means there is a probability. So actually answer already given in the question. That means if there is a probability, then 8 we know that it will be hetero. One will be capital letter G, other one will be small letter g. So based on the question, if there is a probability, then 9 will also have to be hetero. That means it will be capital letter G and small letter g. But if in the question it is written that there is no probability of this child having galactosemia, then it will be capital letter G and capital letter G. That means 9 it will also be hetero, capital letter G and small letter g, and the probability is 1/4. If we do the cross with the capital letter G of square, capital letter G and small letter g of square, and the circle of capital letter G and small letter g, if we do the cross, then we will found it is 1/4 or 0.25% or 0.25, 0.25, or we can say 25%. Definition. Question number 3b1. What is meant by the term mutation? A mutation is a change that occurs in our DNA sequence either due to mistakes when the DNA is copied or as a result of environmental. The next question is number 2. The mutation for galactosemia is harmful, but some mutations can be beneficial. Describe one example of a beneficial mutation. So you can say many. So there's a beneficial mutation often giving antibiotic resistance in bacteria. You can say this answer. Okay, it's very easy. Number C. Some inherited conditions can be fatal, but medical treatment is often available. Explain what would happen to the frequency of alleles for these inherited conditions if medical treatment was not available. So you can say over here that if there is no medicine, then the people won't survive. and there will be less people. Day by day the population will decrease due to this disease. As a result, there will be no reproduction. The people won't survive to reproduce and pass their alleles to the next generation. It's very easy. Next question. Number 4. A. A student reads the following statement. The father determines the sex of a baby. Explain why this statement is true. It is because, you see, male gametes or sperm carry X or Y chromosome, and female gametes or eggs carry an X chromosome. Therefore, the father has to give either an X or Y to the baby, and if it is X, then the baby is female, and if it is Y, then the baby is male. Therefore, it depends on father whether the baby will be male or female. Now let's go to the next question. 4b. The diagram shows sperm cells viewed using a microscope from two different men, man A, man B. Use the information in the diagram to give two reasons. We have to give two reasons why man A is more fertile than man B. In the figure, it is seen that man A has more sperm than man B. It is one of the reason. Another is, and maybe there's some kind of genetic problem here, because there are multiple heads and multiple tails in man B. It can be another reason. Next question, C1. The graph shows how smoking may affect the movement of sperm. Percentage of sperm they are showing, smokers and non-smokers. The average mean sperm count in the semen from non-smoker is 50 million in 1 centimeter cube. That means for the non-smoker, in 1 centimeter cube, 1 centimeter cube is in the graph. Mean in real 1 centimeter cube means its average it is 55 million. Calculate the number of moving sperm in 1 centimeter cube of semen from the non-smokers. So in the graph we are seeing that it is 45% non-smokers, and so to calculate it will be 45% of 55 million. That means 45% into 55. 45 divided by 100 into 55, the answer will be 24.75. Use the information from the graph to explain why smoking could affect male fertility. Answer will be, this is the answer of C number 2. The graph shows fewer moving sperm, which reduce the chance of a sperm reaching and fertilizing an egg in the oviduct, and therefore fertilization doesn't occur because there are fewer chance. Number 5. In a survey, people of different ages were asked if they thought that animal cloning is a good idea or a bad idea. The table shows the results of the survey. So we are saying they have given age group, percentage of age group, good idea, bad idea. Under 24, 10.6, bad idea 89.4. Now 25 to 34, 14.2, and bad idea 85.8. Over 55 it is 23.5, good idea, and in bad idea 76.5. Plot a bar graph to show the data in the table. I think you will do it, you the guys can do it by yourself. The bar graph is very easy. And next question is number B. Describe the relationship between age and what people think about animal cloning. Now, as people, we are seeing from the chart that as people get older, more think cloning is a good idea. Now, 5C. In the 45 to 54 age group, 18 people think that cloning is a good idea. Calculate total number of people surveyed in this age group. Show your working. So suppose total number of people is X. Okay. So in 45 to 54, if we see the chart, it is saying that 15% they are thinking that it is good idea. So you can write how you will solve it. 15% of X is equal to how many people they had given? Total number of people is 18. So 15% of X is equal to 18. Now 15% means 15 divided by 100 into X is equal to 18. Now find the value of X. That will be the answer of your question. Very easy. It will be 120. Some people in the survey did not know anything about cloning before they made a decision. Describe the process of cloning an adult animal using a named example. Now, Dolly the sheep was the first mammal to be cloned. She was born in the UK in 1996 and died in 2003. She was produced using the nucleus from an UDERSTAND, although other cells such as skin cells may also be used. Now the method for adult cloning is, number 1, the nucleus is removed at first from an unfertilized egg cell. Number 2, the nucleus from an adult body cell such as a skin cell is inserted into the egg cell. Number 3, an electric shock stimulates the egg cell to divide to form an embryo. Number 4, these embryo cells contain the same genetic information as the adult skin cell. Number 5, when the embryo has developed into a ball of cells, it is inserted into the womb of an adult female to continue its development. But here in this experiment we have to remember that there will be two sheep. From one of the sheep, suppose sheep A, and the other one it will be sheep B. From sheep A we will remove the nucleus, and from the sheep B, X cell taken from sheep B, and we will only take the X cell by removing the nucleus from the sheep B, and we insert the nucleus of sheep A into the X cell of sheep B. Then later on it will be an embryo cells. It will contain the same genetic information, and later we will insert it into the host mother. Fused cell then develops into embryo, which is placed in uterus of host mother. I think the whole process is clear to you. We are going number 6. Okay, I want to say you one thing, that all the answers have been given in a sheet. Please check the sheet. It will be found in a sheet in a YouTube channel. Please check the answer sheets so that it will be easier for you. You can directly copy the answers and you can check the answers with the questions, so that it will be easy. Please check the answers of each questions. It has been written broadly, elaborately. Okay, let's go to question number 6. The amount of light falling on plant leaves is an abiotic, non-living factor. As light is non-living, we know that affects photosynthesis. Give the balanced chemical equation for photosynthesis. So what are the balanced equation? It's carbon dioxide plus water, which will give glucose and oxygen. So you have to balance the chemical equation. It will be 6 carbon dioxide, 6 water, and 6 molecules of oxygen, and C6H12O6, it will be glucose. Now next question, number B. The leaves of plants that live in the shade, low light, are different to the leaves of plants that live in full sunlight. In full sunlight it is different. Number 1, suggest why leaves from plants that live in the shade are darker green than leaves from plants that live in full sunlight. Here what they want to say, the leaves of plants which live in the low light, means which are getting low light, are different from the leaves which are getting full sunlight. And the question had been said, suggest why leaves from plants that live in the shade, the leaves which live in the shade, they are darker green than the leaves which live in full sunlight. The leaves which live in the shade, they are darker green. Now why they are darker green? It's question number 6B1. In the shade, they are in the shade leaves, that means they are darker green, that means there are more chloroplasts. The answer will be, in the shade more chloroplasts is needed in order to absorb as much sunlight as possible, and the chloroplasts are closely packed, which represents on the upper side of the leaf. The answer is given on the sheet. Check the sheet. So now number 6B, number 2. Explain why leaves from plants that live in the shade are thinner than leaves from plants that live in full sunlight. The answer is, thin leaves have a shorter distance as it is thin for light to penetrate to reach the chloroplasts, thus maximizing light absorption. But the leaves which are in front of the light, in front of sunlight, they are much thicker, because light there, one of the reason is for the sunlight. Okay. Now number C, the rate of photosynthesis changes during the day. Explain the factors that affect the rate of photosynthesis in the early morning. So we know that in the early morning, first we have to find out that which is the limiting factor. So in the early morning, actually light and temperature are the limiting factor. As it is in the morning, temperature will be low, and also the sunlight, it will not be full sunlight. Full sunlight will be in the afternoon. So what will be the answer? The answer will be C1. Answer will be, in the early morning light and temperature levels are low, so stomata will not be fully open, and for this reason photosynthesis will be slow. Next question. Explain the factors that affects the rate of photosynthesis in the early afternoon. So first we have to understand what are the limiting factors in the early afternoon. In the early afternoon, the limiting factor is carbon dioxide. So what will be the answer? In the early afternoon, light and temperature levels will be higher, stomata will be fully open, and so photosynthesis will be faster. For higher temperature, enzymes involved in the reaction of photosynthesis will work faster. For higher temperature, because whenever temperature increased, chemical reaction also increases, so enzymes work faster. However, here the limiting factor in the afternoon will be carbon dioxide. You can check the answers from the sheets which I have given. Check the answers so that it will be easier for you. Next question. Describe how you could compare the rate of photosynthesis in two different plant species. Now here we are comparing two different plant species. So the two plants which we will take over here is Alodia and Cambomba. Now let's see how we will do the experiment. Now I am saying the answer, number 3. We are going to have two different types of plant species. Oxygen can be measured by counting bubbles a given time evolved from pondweed, such as Alodia and Cambomba, as a measure of the rate of photosynthesis. So oxygen will come out, okay, oxygen will come out as a bubble, so we can count the number. To do this, place Cambomba pondweed in an upside down syringe in a water bath connected to a capillary tube. You can also use this for Alodia. Put the weed in a solution of sodium hydrogen carbonate solution. You can then investigate the amount of gas produced at different distances from a lamp. So it's very easy. These are the experiment. The answer is being given on the sheet. Please check the sheet so that it will be easier for you. Now I am going for the next question, number 7a. Question number 7. A student uses this apparatus to investigate the effect of nitrate ions on the growth of plants. So here the student is investigating the effect of nitrate ions on the growing of plants. What effect will it have on the plants? A young plant is grown in a sterile condition containing all the mineral ions needed for growth. The student repeats the experiment with other young plants. The student also carries out the experiment with young plants grown in a sterile condition that contains all the mineral ions except nitrate. That means in one condition there is all the mineral ions needed for the growth, and in another experiment there is all the mineral ions but no nitrate ions only. The student measures the length of the stem. Then it measures the length of the stem of each plant every 5 days. Some of the students' results are shown on the table. Here we can be given time, has been given time in days, average length of the stem in millimeter, average mean. Solution contains all mineral ions, solutions without nitrate ions. So in 0, where the solution contains all mineral ions, at first it is same, 23 and 23 in 0 days. After 5 days, all mineral ions it is 30, nitrate ions decreased, it is 25. After 15 days, all mineral ions is 45, nitrate ions it decreases, it's 30. And gradually the solution which contain all mineral ions, it after 25 days it is 98. That means it increases, but in case of nitrate ions it is 38. It also increases but at a slower rate than mineral ions. Then after 35 days, where there is all mineral ions, it is increases in size, but after 35 days in solution without nitrate ions, it does not increases so much. Describe the growth of plants in each solution. Now we have to describe the growth of plants. So 7a, the answer will be, plant grew much more in the solution containing mineral ions compared with the solution without mineral ions. But in case of mineral ions, it decreases after some days and growth stops at 45 days. And without nitrate ions, it stops growth after 25 days. Now B, explain why young plants absorb more mineral ions than air is bubbled through the solutions. It is because the oxygen is used for aerobic respiration to provide energy required for active uptake of the ions. You can check the answer sheet. Answer has been given in the sheet. Now let's go to the next question, 7C1. Now we will question 7C1. Suggest why each solution is sterilized at the start of this investigation. So the answer is, the solution was sterilized so as to prevent microbial, especially algal growth. These are the microorganisms, algal growth, that may absorb the mineral ions which might otherwise affect the growth of the plants. So here actually there are microorganisms, it may be algae or bacteria also. So they may absorb the mineral ions and it will affect the growth of the plants. Answer have been given in the sheet. Please check the answer. Next question. Number 2. Suggest why the glass jar is surrounded by cardboard during this investigation. Because it is to exclude light so that it prevents the growth of algae. Otherwise algae, with the help of light, it can make food and it can grow. So the answer will be to exclude light so that it prevents the growth of algae. Next question. D1. Identify the dependent variable in this investigation. And here in this investigation the dependent variable is length. Number 2. Name one biotic living variables that should be controlled in this investigation. It will be mass of plant. Number 8. The diagram shows the distribution of two plant species in a small area of a field. There are two plants, one is bunch grass and the other one is plantain. A student uses a square metal frame to help count all the plants in the area. What is the name given to the metal apparatus that the student uses? So we know that the name is quadrant. Quadrant because as it is 4 shaped, so we call this as a quadrant. Number B1. He counts the number of plants in all of the squares he marked out in the field. Complete the table to show the number of plants of each species and the average mean number of plants per meter square. So first one have done already. Here species is bunch grass. There are 7 plants over it. Now average number of plants per meter square, that means per meter square how many plants, what will be the average number of plants. I had shown this in a sheet how you will solve this paper. Just check the answer sheet. Say about 36 meters square it represents 7 plants. So we can say that for 1 meter square it will be 7 divided by 36 is equal to, answer will be 0.19. Similarly for 36 meters square it will represent 8, and for 1 meter square 8 divided by 36 it will be 0.222. So this is the answer. Let's go for next question, number 2. Frequency is another measure that can be used to study distribution. Frequency is the number of squares that contain at least one plant of the species being counted. This value can also be expressed as a percentage of the total number of squares sampled. Complete the table by giving the frequency and percentage of the total number of squares sampled for bunch class. It's very easy. See, actually, if you see the table, plantain have been given. The frequency is 8, percentage is 22. So with this reference we can also find the frequency of bunch class and the percentage. As plantain it is 8, we can say that for 8 it is 22%. For 1, how many it will be? 22 divided by 8, and for 4 it will be 22 divided by 8 into 4. The answer will be 11%, and the answer will be 11. Next question, number C. Describe how the student could estimate the population size of plantain in a very large field. So the answer is given in the sheet. Check the answer. It is randomly place quadrant several times on the playing field. Count the plantain plants in it each time by putting the metal grids down in random areas and counting the number of plantain under the grid. After that we can then calculate the average and then multiply this by the area of the field. Let's go for the next question, number 9. The diagram shows a cell from the human nervous system. Here it is given A, B, C. Name the structure labeled A. So A is nucleus, A1. Number 2, A2 is axon. Number 3, A3 is arrow from the cell body towards the dendrites. Check the answer sheet. In the answer sheet, the answers have been given. Number B. Describe the role of this neuron in a simple reflex arc. It's very simple. The role is to transmit nerve impulses from the central nervous system to the effector. Effector can be muscle. Okay. Next question, number C. Nervous communication differs from hormonal communication. State 3 ways that nervous communication differs from hormonal communication. It contains 3 marks. It's very easy. Check the answer sheet. It is also given in the answer sheet. All the answers has been given in the answer sheet. So answer is, the nervous system uses electrical impulses to send signals through neurons, whereas the hormonal system uses chemical messengers transported into blood plasma to target cells. Nerve impulses can travel much faster than hormones. Next question, number 10. The passage describes how different organisms are classified into groups. Complete the passage by writing a suitable word or words in each of the spaces. Plants are multicellular organisms. They have chloroplasts to carry out photosynthesis and cell walls made of cellulose, because cell wall is made up of cellulose. They store carbohydrate as a starch. In plants, they store carbohydrate as a starch or as sucrose. Animals are also multicellular but do not carry out photosynthesis. They are able to move from place to place and are always described as consumers in food chains. They store carbohydrates as, we know that it's very easy, as glycogen. It will be so. Next para. Bacteria are single cell organisms. They do not have a nucleus. Instead, they contain circular plasmids and smaller circles of DNA called, they do not have a nucleus. Instead, they contain a circular chromosome and smaller circles of DNA called plasmids. Most bacteria feed off other living or dead organisms, but some bacteria can make their own food by photosynthesis. Examples of bacteria include Lactobacillus used in the production of yogurt from milk, and Pneumococcus that acts as a pathogen causing the disease pneumonia. So this questions is finished. Now we are going for number 11. And one thing is that all the answers have been given in the sheets. Please check the sheets with the questions, then it will be easier for you. Now let's go to the question number 11. The photograph shows the logs left behind after an area of forest has been cut down. These logs are decomposed by fungi. Describe how fungi decompose tree logs. Fungi use saprophytic or saprotrophic nutrition. They secrete enzymes such as amylase, which break down, or you can say digest, the log into soluble products such as glucose, and they do a process. After that, there is a respiration process, which are then absorbed by the fungi via active transport. And here we have to remember that this is extracellular digestion. Here the digestion which takes place, it is extracellular. The answer has been given in a sheet. Please check your answer. Now let's go for the next question, number B. Some of the logs removed from the forest are used to make garden benches. The photograph shows a garden bench. The garden bench is painted with a fungicide solution. This prevents the wood being decomposed because fungicide kills bacteria. So here is a bench we are seeing in the picture. This had been, in this bench they had given one solution which is called fungicide solution. There are different fungicides that can be used. Design an investigation to find out which fungicide is best at preventing the decomposition of wooden logs. So we will test this with different kinds of fungicide solution in this experiment. Then we have to find out the result, which one is better. Your answer should include experimental details and be written in full sentences. Answers have been given in the sheet. Please check from the sheet. Now the type of fungicide, which is dependent variable, will measure the decomposition of wood logs. So the way I would do it by measuring the weight of wood before and after the experiment. We need a particular time frame for doing this test, and we also need lots of control variables, such as wooden logs will be from same tree, they will be the same age, and the temperature will be the same during the experiment. So these are the questions which we have solved. No more left. And again I am saying, please check all the answers of the questions from the sheet. In the sheets, answers are available, so it will be easier for you, and the answers have been written elaborately. Okay, thank you very much.